Hi, I'm Dr. Alice Akinyele, and I will be covering the topics of early embryology. By early embryology, I mean that we will discuss what happens in the first 4 weeks of the life of an embryo. And this involves gametogenesis, the formation of the spermatozoa and the oocytes in the first place, fertilization, the joining of the sperm and the ovum to form the zygote, cleavage, the process of cell division during the first several days, followed by implantation of the embryo into the wall of the uterus. Now that embryo forms into a 2-layer embryo, and then a 3-layer embryo, and then the body folding happens to develop into the adult body structures. Development of the different organs after that will be covered separately with each organ system, so you can make the connections between the embryology and the gross anatomy of that organ. We can't talk of the embryo without first talking about how the embryo is created, and that starts with the male and female gametes, the sperm and the ovum. The process of creating these spermatozoa and oocytes is called gametogenesis. By the end of this session, you will be able to explain the process of gametogenesis, and that includes spermatogenesis, which is how spermatozoa are produced, and oogenesis, or the formation of the oocytes, all the way through ovulation and fertilization. I also expect you to be able to predict a few chromosomal abnormalities that could occur as a result of disruptions to gametogenesis. Now, the sperm is a germ cell, or gamete, and contains half the number of required chromosomes, so 23, or it's haploid, instead of the typical 46 diploid chromosomes in most of the cells of your body. Now, gametes end up with 23 chromosomes through a process of division called meiosis. This is a type of cell division that occurs during gametogenesis. In males, this process is called spermatogenesis, and it begins in the testes. Spermatogonium, seen here, or spermatogonia plural, are the progenitor cells that transform into the primary spermatocytes. This then undergoes the first meiotic division to form the secondary spermatocytes with half of the genetic material. The secondary spermatocytes then undergo secondary meiotic division, duplicating into 2 spermatids each, such that one primary spermatocyte after the first and second meiotic divisions result in 4 spermatids. There are 2 types of male gametes, obviously, one that will end up with the X chromosome and another that will end up with the Y chromosome. So 2 types when we think about the sex chromosomes. The final step of spermatogenesis is called spermiogenesis, which is the maturation of the spermatids into actual sperm. The process of spermatogenesis takes about 2 months. So someone knocks out all your sperm, 2 months later, you'll have more. Let's compare that to oogenesis, which is the process through which oogonia, primordial oocytes, are transformed into the primary oocytes. This maturation process begins during the fetal period and are transformed into the oocytes. So when we look at the ovary after birth, all the primary oocytes have completed the prophase, that first stage of mitosis, of the first meiotic division, and they remain in prophase until puberty. And that's why our image showing oogenesis doesn't begin with oogonia, because at birth a female already has all the primary oocytes they will ever have. Actually, most of the oocytes in a newborn do not even make it to puberty. And out of the 40,000 that remain at that point, only a few hundred will ever be used at all. So do the math. Once a month, 12 months a year, from the age of 10 to about 50 or 55, this means that every ovum being released by an 18-year-old girl is actually an 18-year-old primary oocyte. And every ovum being produced by a 35-year-old woman is actually a 35-year-old primary oocyte. Food for thought, isn't it? Now, shortly before the release of the oocytes into the fallopian tube, a process known as ovulation, the primary oocyte snaps out of this long slumber that it has been in prophase for and completes the first meiotic division. Now, unlike in spermatogenesis, the cytoplasm doesn't split evenly between the 2 daughter cells. One receives almost all of the cytoplasm, and this will form the secondary oocyte, whereas the other daughter cell is called the first polar body, receives very little, and will therefore degenerate after a short time. At ovulation, the nucleus of the secondary oocyte begins the second meiotic division. This secondary oocyte is quite large, surrounded by a covering of amorphous material, the zona pellucida, and a layer of follicular cells called the corona radiata. The secondary oocyte is fertilized by the sperm, and the second meiotic division is completed, and a second polar body is then formed. So to recap, spermatogenesis takes about 2 months, while oogenesis begins during fetal life and does not actually end until ovulation. The final stage of spermatogenesis, when the spermatids transform into 4 mature sperms, is a process known as spermiogenesis. And now during this metamorphosis, condensed nucleus forms the head of the sperm. The Golgi forms a membrane-bound vesicule, the acrosome, which caps the head. The centriole initiates the assembly of microtubules in the tail for movement, while the residual cytoplasm is shed. Now, the mitochondria actually gathers in the middle piece, and you can think of this as an engine that powers the movement of the sperm, which is why it's arranged around that tail. Now, gametogenesis involves meiosis, which consists of 2 meiotic cell divisions to result in germ cells with a haploid number of chromosomes. When this division is impaired, we end up with chromosomal abnormalities. Homologous chromosomes line up and then migrate along the meiotic spindle during the meiotic division. One thing that could go wrong is that they simply do not separate. This is called non-disjunction. As a result, some gametes will end up having 24 chromosomes and others only 22, instead of both having 23. If a gamete with 24 chromosomes combines with a normal gamete with 23 chromosomes, you will have an extra set of chromosomes. This is called a trisomy. And you would think that if 2 is good, 3 is better, but it's not. The most well-known trisomy is trisomy 21, or Down syndrome. Here, there are 3 chromosome 21s instead of just 2. And this genetic disorder results in developmental delay, cognitive impairment, craniofacial abnormalities, and a constellation of associated conditions such as congenital heart defects, hematologic problems, endocrine abnormalities, among others. Now, if the gamete with 22 chromosomes combines with a normal gamete, it will result in a 45 chromosome zygote instead of 46. This condition is called a monosomy. Because only one representation of a particular chromosome pair is present, most often a monosomy is actually incompatible with life and will result in a miscarriage. The most common viable monosomy is the monosomy of the X chromosome, which results in Turner syndrome. And you'll learn a lot more about these in genetics.